Hello, everybody. Thank you for the kind introduction, Anais. I'm going to give you today an overview of the work that we're doing in the team, which is focused on the development of multimodal learning methods for the integration of single-cell data. But first of all, the motivation for this. Since 20 years ago, single-cell sequencing technologies have been introduced, and since then, a lot of different data modalities can be measured at the resolution of the single cell, going from gene expression with single-cell RNA-seq to DNA methylation, cell attack, and even spatial information with spatial transcriptomics and spatial proteomics. Now, all these different data types provide complementary information on the cell. So it's like if you're looking at the same object from different viewpoints. And it is from this complementarity that comes the interest to be able to jointly analyze all these different data in order to better understand our cells. And what we focus on the team are methods that are called multimodal learning methods that can jointly analyze and jointly learn from different data modalities in order to take advantage of this complementarity and better understand the state of our cells. So in practice, we see these different data sets as high-dimensional matrices, and we can either focus on the observations that are the cells, or the features that are the different molecular entities that we are measuring, for example, genes in gene expression or peaks in chromatin accessibility. If we focus on the observations, we develop multimodal dimensionality reduction methods. So here the objective is to be able to jointly embed cells from different data modalities in order to better understand what are the cell types and the cell states that are present in the data. Or we can focus on the features and develop multimodal molecular network inference methods. And here the objective is to identify the interactions in between molecular entities that can determine the difference in between one group of cells and another group of cells. So we start from the first direction. And for the first direction, the first problem is to understand how to study similarity in between single cell profiles coming from different cells. And for this, in collaboration with Gabriel Perrier, that is a mathematician in NES, and in the context of the PhD work of Gert Jan, we have been proposing optimal transport as a metric to study similarity in between cells. So what we are doing is to see single cell profiles coming from two cells as probability distributions, and then we are measuring their optimal transport distance as the minimal effort needed to morph the distribution, the expression distribution, for example, of cell A into cell B. So mathematically, we want to identify the transport plan P, given a predefined that optimizes this, let's say, distribution morphing, given a cost C, that is the cost that is imposed in the beginning, and that will tell us how much cost we pay in order to move a unit of gene expression from one cell to the other one. The idea biologically behind it is that cells that belong to the same cell type should have activated more or less the same biological processes, and so it should be easy to morph the distribution of one into the other. So we then benchmark the method, and I will show you one of the applications that is on real data. Here we have different data modalities, single-cell RNA-seq, single-cell attack, and single-cell methylation. These are the performances based on adjusted rand index on data for which we know the true groups that are present in the data, the true labels of the cells. And these are the performances that you achieve with a state-of-the-art method, which means PCA dimensionality reduction, building a K-NN graph in between the cells based on Euclidean distance, and then applying community detection on the obtained network. This is the improvement that we observe if we just substitute on the same procedure optimal transport in place of Euclidean distance. And it was interesting to see, for example, in the second data set of colorectal cancer NOLMAR, where you have a super big population and small rare populations, that with optimal transport, we managed to identify some of these rare populations, while with the Euclidean distance, we would just be able to well identify the epithelial population that was the most common one. So encouraged by this work, we moved to combine optimal transport for the integration of multi-omics data, and we designed MOGLI, which is combining integrative non-negative matrix factorization with optimal transport. The idea here is that we have different data profiled from exactly the same set of cells. So it's 10x multiome or site-seq, RNA and attack from the same set of cells. And what we do is to write them as a product of lower dimensional matrices, and in this way embed them into a shared lower dimensional Latin space. Differently from a standard integrative non-negative matrix factorization, here in the loss we use optimal transport instead of Euclidean distance, and we minimize the difference in between the original matrix and the product of H times W. What we can do with it? We can use the matrix W that provides a lower dimensional representation of the data in order to plot our cells, identify clusters of cells, populations, and so on, based on different data modalities. And we can focus on the matrices H that are the loadings in order to interpret biologically the space and know what are the genes and the motif that are representative of the different latent dimensions. So we extensively benchmarked the methods in terms of cell embedding, showing that it was performing comparably to the best state-of-the-art methods. So it was not super interesting on this side, but the interesting part comes from the biological interpretability. And we tested this on PBMC data profiled for T-seq, which provides 3 omics from the same set of cells: attack, surface proteins, and RNA. And then we applied 3 factorization methods that were the only methods in the state of the art that were providing a biological interpretability together with the output. So Mowgli, that is our method, MOFA plus, and standard NMF just to have a baseline. And what we want to see, what we are plotting in this last plot on the right, is here, I don't know if you can see it, is the average, let's say, weights for a factor in a given cell type versus the average age, the average weight on the other cell types. So in principle, if we have a method that is specifically associated to the biological signal of one cell type, it should get just 1 factor on the top left and all the others on the bottom right. These are the results that we observe. I'm showing it for 3 cell types just for simplicity. And you can see that in the top we have Mowgli, and we have clearly 1 factor, 1 cell type association, for example, factor 7 for erythroid cells. For MOFA, this happens sometimes, for example, factor 0 in B cells, but sometimes you get multiple factors associated to the same population, and these do not reflect subpopulations of, for example, erythroid cells, but really factors that are well spread across the different cells. And then for NMF, this is even worse, and it's a known characteristic of NMF. Then just to show you an example of application, here you have the embedding that we can get with Mowgli. And you have a broad annotation of the cells, but actually you can get more. And just to give you an example, based on 2 factors, we can identify a factor that is active on the upper part of the B cell cluster and a factor that is active on the bottom part of the B cell cluster. And based on the markers, we can annotate them and know that the top 1 is memory B cells and the bottom 1 is naive B cells. Now, all this works if the data are profiled from the same set of cells. But sometimes you want to integrate data that are coming from different cells. And this is more challenging because you cannot use anymore the cells in order to bridge the information across them. And at the same time, you don't want to convert the features of one data set into those of the other one, because you would move from tens of thousands of features sometimes to hundreds, and that's not good. So what we have been doing, and that's the PhD work of Jules that is presenting also in a poster in the conference here, so I invite you to go to visit this poster, is to develop SC confluence, which is based on regularized inverse optimal transport. So the idea here, first of all, is that, again, we want to reduce the dimensionality of our data, and we do it using autoencoders. So we have 2 data sets, for example, single-cell RNA and single-cell attack, and with autoencoders, we project them into a lower dimensional lattice space. Now, the issue is that if we only do this, we are just having the 2 data sets of lower dimension, but we don't learn how they are related to each other, and so it's not a proper integration. And to realize a proper integration, it's where inverse optimal transport comes. So what we are doing is to use a translation of the second data set, for example, attack, into a form that is similar to the first, gene activity in this case. And then we use this information in order to define a plan that we can use to mix the cells in the obtained latent space. So you see that we have an addition of a loss based on inverse optimal transport. Then we benchmark it with the state of the art. We have different metrics. The first one and the third one will measure to which extent we are preserving cell types. The second and the last one are measuring to which extent we are putting together cells that we know that are the same across the 2 modalities. Because we are using to evaluate the methods pair data, we are not giving to the methods the information that the cells are the same, and we want to see whether they are putting them close to each other in the latent space. And what we can see is that in the first data set that is Multiom, SC confluence performs close to the state of the art, I mean, as the best performing state of the art, but more interestingly, in the context of site-seq, we can see an advantage. And this is probably coming from the fact that when you have little features on one data set, the fact of not being forced to convert the features of one data set into the other one brings an advantage in terms of quality of the latent space that we are building. And I will show you now two examples of applications that we have been testing on. The first one is integration of single-cell RNA and SNFish data from mouse cortex. So here, single-cell RNA-seq is a throughput, but you don't have the tissue localization information. SNFish, you measure way less genes, but you have the tissue information and you have the tissue slice. We can integrate them, and here you have the UMAP visualization of the obtained integration with a circumference, but then we can play a bit with that. And once integrated, we can try to hide some of the genes that are measured in the SNFish experiment and try to see whether the method can predict the right spatial expression. And here you have an example with 3 genes. On the top, you have the ground truth expression, and on the bottom, the imputation done by the method. And it's nice to see that the overall patterning is well represented in the imputation with respect to the ground truth, with some exceptions. For example, in the case of the second gene, there is an area that is supposed to be downregulated that we are not capturing with the method. So there is still space for improvement in this sense. And then we could also go to some genes that are measured in the single-cell RNA-seq and not present in the SNFish, and try to see whether they could have a pattern in space that is relevant and similar to the markers that were measured in the SNFish. And we could identify some of the genes that were having this interesting patterning. The second application is instead related to integration of morphologies of neurons and single-cell RNA-seq expression data. So here we work on mouse primary cortex, and we work on patch-seq data that provide at the same time single-cell RNA sequencing and images of neuron morphologies. And the objective is to combine together the expression and the morphologies of the neurons. We have one part of the data set where only single-cell RNA-seq is available, and we have one part where both single-cell RNA-seq and morphologies are available. We use the single-cell RNA-seq in the second data set to bridge the information in between the morphologies and the single-cell RNA-seq of the first one. This is the embedding that we get with the circumference. So you can see that there is quite a good mixing in between the data modalities, and we are preserving the different populations present in the data. But their positioning is more spread. It seems that we are detecting more heterogeneity inside each population with respect to what was possible with single-cell RNA-seq. And this is an information that is coming from the morphologies. So we can see, for example, that focusing on IT and DT neurons, we are actually detecting a variation inside them that goes from more untouched to more toughed neurons. So this heterogeneity is representing real morphological features. And then we can also play on the connection in between the data and find, for example, that there is a transcription factor that seems to have its expression moving across the same manifold of the morphological variation. And we could validate that this transcription factor is known to be involved in the morphological features through calcium channels regulation. So for the conclusion of the first part, we have shown that optimal transport is good for detecting cell-cell similarity in different single-cell omics data. We have proposed Mowgli for paired multi-omics integration that is particularly good at the level of the biological interpretability of the factors. And then we have proposed the method, which is called SC-confluence, for the integration of unpaired single-cell data. Both methods are available on the GitHub of the lab and well documented, and I invite you to test them out. Then the second direction is to focus on the features and to reconstruct molecular mechanism. And for this, we propose Humus, which is based on networks. That's the PhD work of Remy. So here what we are doing is through multi-layer networks, we are integrating different networks obtained from different single-cell data. So you can reconstruct a network in between transcription factors based on protein-protein interactions, a network from single-cell attack based on peak cooperativity, a network from single-cell RNA based on co-expression in between the genes, and then you can infer interactions across the nodes of the different networks based on biological knowledge, motif analysis in the first case, and peak proximity to the transcription starting site of the gene in the second case. And what's the advantage of this type of approach in respect to others that are based on regression analysis is that you keep the full richness of the information present in each data set. This means that you can take into account cooperation in between biological macromolecules that you are missing with the regression analysis. You can easily include other omics data if you know how to relate the nodes of these other omics data set to the one of those already present in the network. And it doesn't require to work with paired data. At any moment, you need to pair your single-cell data. Then what's the output that you can get? What you can do is to run random walk with restart and you can get different type of predictions. You can predict the targets of a transcription factors, you can predict the enhancers associated to the gene, all depending on where you start your random walk with restart. Then we benchmark it, and I'm going to show you just one subpart of the benchmark focused on transcription factor target predictions. So we run humus and other state-of-the-art methods, and we compare them with a ground truth that is based on intersection in between perturbation experiments and ChIP-seq. We take into account intersection to avoid as much as possible false positive interactions. And these are the results that we get. It's 4 different data sets, human embryonic stem cells and mouse embryonic stem cells. What we are plotting on the y-axis is the average number of true targets of a transcription factor. When cooperation in between the transcription factor is taken into account, the orange line corresponds to humus without cooperation of transcription factors. Given that the other state-of-the-art methods do not consider cooperation in between TF, we wanted to test both types of results. And overall, we can see that we perform better over the state-of-the-art. It's interesting to see that in the last data set, where you were forced to pair the data, the other methods drop in performances. And the black line that corresponds to random, all the methods are quite close to the random performance. But overall, it has to be noticed also that transcription factors tend to have hundreds of targets. And here we are getting on average 8, 10 true targets, indicating that there is still a lot of space for improvement. And then just to challenge a bit the method, we used it to integrate, to reconstruct regulatory mechanism from single-cell RNA, single-cell attack, and methylation data profiled from different cells. And this means adding an additional network based on methylation and linking it to peaks based on overlap in the genome and to genes based on closeness to the transcription starting site. And then we use the regulons obtained by this network, meaning the transcription factor and their set of predicted targets, to reconstruct the heterogeneity in between the cells that was already observed at single-cell resolution. So using here hundreds of regulons, we can reconstruct all the heterogeneity that could be observed from the use of 20,000 genes in normal single-cell RNA-seq data. And then we could focus on the 5 more strongly associated transcription factors to each population, and we could find that many of them were already documented and validated to be associated to that regions or to neurons or brain. And interestingly, those indicated with the black arrow are those that we would have lost if we were not considering methylation, indicating that methylation is an important feature to take into account, not surprisingly, and that it's relevant to have methods that can easily adapt to a lot of different omics. Now, as I said before, the existing methods for building single layers don't seem to do great, and there is still a lot of space for improvement in the inference of gene regulatory network. And that's for this reason that with Jeremy Calfon, that is also a PhD student in the lab that is talking this afternoon about this work, we developed SPRINT, which is a foundation model focused on the inference of single-cell GRN. So it is basically a bidirectional transformer that is taking in input gene expression values, summarizing them into 3 main tokens. The first one is the gene ID that is here converted into ESM2 embedding in order to be able to possibly generalize to other species that are different from the ones of which we have been training it. And then the gene location to know whether 2 genes are close to each other in the genome and possibly can co-regulate each other. And then the expression values of the genes. And then we are training it on the cell-by-gene data with 50 million cells from different species, human and mouse, and different diseases, on 3 main pre-training tasks. The first one is denoising. So we want the method to learn that in single-cell RNA-seq there are dropouts. There are different technologies that are bringing different types of noise depending on the sequencing technology. So the method should be able, once you give it a noisy profile, to be able to reconstruct the actual real profile of the single cell. Then bottleneck learning, we want the method to be able to compress the information of the gene expression data and then reconstruct it. Be able to reconstruct an expression, let's say, profile that is similar to the starting one. And finally, classification. We have a lot of annotations on these 50 million cells, going from cell types, tissue, disease. We want the method to be able to recognize them and to be able to understand what's the difference in between different states. And what are the type of outputs that we can get? We can get label prediction based on the fact that we are training on classification. We can provide embeddings and batch-corrected embeddings. So we can denoise expression data also based on the training. But more importantly, behind all these, there is the fact that the method is learning what's the model behind the functioning of our cells, ideally, and so what are the interactions in between the genes that are important to do these predictions. And so averaging the attention matrices that you get from the model, you can obtain the gene regulatory network is associated to the cells. And just to give you an example of output, here you can see an example of UMAP representation after batch correction with the annotation of the different cell types and the GRN associated to each different cell type. So we have been benchmarking it with respect to the state of the art, obtaining, let's say, interesting performances. I'm not going to show you these results, and I invite you to go to the presentation of Jeremy this afternoon to know more about this work. So to conclude on this part, Humus seems to provide better performances in GRN inference if you are interested in combining information from different single-cell data modalities. Still, at the level of the inference of single networks, we don't have ideal performances, and there is a need for improvement. And we propose a solution based on the use of transformers, and we're further working in this direction in order to further improve it. Both tools are available on the GitHub of the team, again, Humus and Sprint, and I invite you, they are well documented and so on, and I invite you to go to test them out. Then last part, all what I've shown until now is based on a static view of cells into groups for which we want to identify regulatory mechanisms. But actually, cells are not static. They continuously modify their phenotypic characteristics in time, mainly influenced by their molecular information and molecular regulation inside the cell, but also based on the interaction with the neighboring cells. So we want to take into account space and time information into the analysis. And that's what we aim at doing with the recent ERC starting grant that we started in spring 2024. And I'm going to show you one first results, which is called Stories, which is a tool that has been jointly developed by Jules and Gertian, and is presented by Jules also at the poster B293, that I invite you to go to see and discuss with him. So the tool is called Spatiotemporal Omics Energies. And what we are doing is to try to infer trajectories, not only considering expression data, but also space information. How we are doing it? We start from input data that are spatial transcriptomics measurement profiled at different time points, for example, different phases of an embryo development. And what the method is doing is to try to learn the differentiation potential that is behind this evolution of cells. So we have a neural network, and here it's a simple one, an MLP, that is learning to minimize the difference in between the prediction that we make of what will happen to our set of cells from time t to time t plus tau, and the actual measurement that we have available time t plus tau. So it requires that you have multiple measurements, because if you have only two states, it's difficult to train the model. And how we are writing this loss and how we are optimizing this different, we are doing it using fused Gromov-Vasses time distance, which means taking into account two major terms. A linear term that is checking that the expression in between the prediction and the actual measurement of the cells is close to each other. And this is what has been already done by other methods that are trying to infer potentials. But then we add an additional term that is a quadratic term that takes into account the spatial component. So we want that cells that are close in the measurement in space are also close in the prediction in space. So we want the spatial consistency with respect to the prediction and the measurement to be emphasized in the method. And what you can do with that, you can infer trajectories, you can reconstruct this differentiation landscape, and more importantly, you can also get genes and regulators that are associated to the different directions of variation of this potential. So you can interpret biologically this potential and know whether it's representing real biological signal or artifacts. And then we went to test it, and the main question was, is really the space bringing important information for development? I'm going to show you one example. We did the benchmarking on 3 different atlases. This is a mouse atlas, and we have our training set and then an early test set where we take another batch profiled on time points that have been already seen by the method, and then a late test set where we take data from another batch on time points that have not been seen by the method. And what we are doing is to try to predict what will come next. And these are the performances that we obtain here compared with respect to prescient that is not using space information. Here you have the vast time distance in between the prediction that the method is making and the actual measurement that is available. And you can see that we achieve lower distance with respect to the ground truth, so a better prediction of the ground truth. And then the transition accuracy that is measuring basically biologists have annotated which cell type is supposed to transition to which cell type in the different time point. And we can test to which extent our prediction correspond to those expected and annotated by the biologist. And you can see that we achieve a higher accuracy, indicating that even here the space is bringing information. And then we went a little bit more in the detail and we focused on mouse midbrain under a specific transition that is from radial glia cells to neuroblast and glioblast. And so you have radial glia cells that we split mainly into two populations going either in one direction, either in the other. And we want to see whether the space is bringing important information in this setting. And so methods taking into account the space are more reliable than those that don't do it. And here you have an example of one of the slices, E16.5. On the left, you can see the annotation of the cell types in the slice. So you have pink that corresponds to neuroblast, green that corresponds to glioblast, and blue, light blue, that correspond to radial glia cells. So radial glia cells are a bit present all over in this layer. And then you will have instead the final states, neuroblast and glioblast, that are well spatially positioned. Neuroblasts are on the top part, then you have glioblast in the middle, glioblast in the end, and a little bit of neuroblast in the middle in between the two. On the right, you have the same slice where we are plotting the potential predicted by the method. And you can see that we are plotting it only for radial glia cells. And you can see that in the top part, most of the radial glia cells will tend to become neuroblast. On the middle, they will tend to become glioblast, as much as in the bottom, indicating that these radial glia cells are not positioned randomly in the mouse midbrain, but their positioning will influence what they will become as a final state, whether neuroblast or glioblast. So with this, I'm going to conclude. I want to thank my team, in particular Jules, that did all the work of SC Conference and stories, then Remy that did the work of Humus, and Jeremy that did the work of Sprint, all collaborators and funding, and I'm happy to take questions.
Yeah, can you, can you show?
Okay. I have a question regarding of the regulatory networks. How do you decide which regulatory network or which signaling network or different interaction network you are using for each of your single cell protein? Can you do some kind of benchmark?
Can you repeat the question
Yeah.
and speak here?
Yeah, so the question was, how do you choose what type of protein-protein interaction network to use in the transcription factor interactions on the top of the multi-layer network? Okay, and for transcription factor regulatory network too. So for protein-protein interaction, we just use string. We didn't test what was the impact of other methods. In principle, that network can be also personalized to the biological setting that you're interested in, and for sure it will give better information than what we did for the benchmark. We tested intersection with respect to ChIP-seq data that are evaluating cooperativity in between TF. And all what was in string was present also in the ChIP-seq data. So that's why we kept everything there. Regarding the gene interactions, what we did was just to apply Gene3 in order to infer interactions in between genes for the single-cell RNA-seq data set. We didn't test what was the impact of other methods. One, and we had seen that it was working better than others. Actually, we did GRN boost for the bigger data set because with Gene3 is impossible, and we use Gene3 for the smaller data set because it's more precise than GRN boost. Welcome.
Okay.
Hi.
Yeah.
Can you provide an intuition for you like in the context of single cell RNA-seq integration, where you're replacing the Euclidean distance with the optimal transport, and you say you get much better results. Can you provide an intuition for why you think that is? And then also, what if I used cross entropy instead of, as a measure, instead of, for example, Euclidean distance? Could I do that instead of the optimal transport computations?
Okay. So for the second part of your question, we didn't test it, so I would not be able to tell you. But for the first part of the question, the intuition of why it's working better, it's because Euclidean distance will compare. So if you have two expression profiles of two cells, you will compare them gene by gene. Now in single cell RNA-seq, you have a lot of dropouts, and this, when you compare a certain value with 0 that is actually a dropout, so it falls 0, this will count a lot in the Euclidean distance. In the case of optimal transport, instead, you're working at the level of distribution, and you allow to move things in between genes that have a low cost in between them, meaning that they have similar expression profiles across the different cells. This means that you can easily detect, even if a gene has a dropout, you will find another gene that has really move to the other location of the cell. I don't know whether I were clear. So working on distributions of gene expression and allowing for moving beans in between genes that have a similar expression across all the cells make you better deal with dropouts with respect to measures that are comparing one by one the expression values of your genes.
And then one line, and then
Yeah.
Also related to this distance measure that you're using, this optimal transport, is it useful to do these predictions for new cell types or something maybe between species? Would that be more useful? Because there's lots of work on that too, like when you train your models on some cell types or species and you want to predict for others. Would that help in that? For that problem?
So
is distance.
You are talking about the trajectory part, so the very last part. The first part, the integration of multi-omics data?
Yes, but using these methods for...
So you mean I have, for example, a single cell RNA-seq data from one species, a single cell RNA-seq from another species, and I want to integrate them together. You want to predict the second one.
Or the cell type that you held out and you want to make prediction of that cell type that you have not trained on.
Okay, so the thing is that, the method is not trained on one set of data and then tested on another one, but it's learning at the same time how these data should be related to each other. So in principle, what you can do is that it's taking into account the complementarity. So if you have one cell type that is present in one data set and not in the other one, the method will put it in a way that is separated from the cell types that are shared with the other data set. So it will not mix it up with the others. But you cannot apply to one data set and then, in principle, you can take an additional data set and try to project it on the embedding that you have obtained with the method. But you cannot, with the method, make predictions of what will happen on another data set.
We have an online question by Zien Kim. Does the flow function take into account the gene expression activity of other cells?
The flow function...
Which
maybe it's the trajectory inference. So there is the linear part that is taking into account the expression of all, is taking into account all the other cells, and then there is the part of the space that is just taking into account the spatial coordinates, and it doesn't care at all about the expression.
So for the sake of time, we have to move on. Thanks again, Laura.
Welcome.
It was great.